Hello dear students, Mr. Poor here, and in this video I am explaining how to investigate the effect of enzyme concentration on the rate of digestion of starch. We know that enzymes are protein molecules that act as biological catalysts. If we remind ourselves that we know that enzymes are very specific in their actions, that's because they have the active site that is complementary in shape to the substrate. So we have studied previously that enzymes do work in different ways, and one model is known as the lock and key hypothesis. So in the lock and key hypothesis, just remind ourselves that the enzymes have a fixed active site and that eventually the substrate fits within this active site just like a key, and the active site here is acting as a lock, isn't it? And once the binding has occurred, the breaking down or the joining of the substrate occurs. So being in the active site, the enzyme basically lowers the activation energy and the reaction proceeds as the products are formed. As you can see, so the products will be different from the reactant, so they no longer fit to the active site and they move away from the enzyme. What is interesting, we consider the enzyme as a catalyst because it remains unchanged at the end of the enzymatic reaction. So therefore the same enzyme can be used over again, and new substrate can bind to its active site in order to be catalyzed and potentially broken down to products. And as you can see, that if the substrate does not have a complementary shape to the active site, the activity, the enzyme catalysis cannot occur. So each enzyme will only catalyze a particular reaction or class of reaction. Okay, let's have a look at this paper here, November 2017. Question paper 6 version 2, and we're going to have a look at question 1. So in this paper, the investigation is about looking at the effect of the enzyme concentration. That is, we're going to increase or decrease the amount of enzyme concentration in the experiment, and we're going to see how it affects the rate of digestion of starch. Now, before looking at this investigation, let's look at what we know so far on how enzymes behave under certain conditions. For instance, you have two graphs here, each one of them showing the effect of either the enzyme concentration on the rate of reaction or the substrate concentration on the rate of reaction. In this particular experiment that is shown in the November 2017 paper, the investigation is focused on the enzyme concentration. So as you can see, as we increase enzyme concentration, that is, we're giving more and more enzymes, the rate of reaction also increases. We mean the rate of enzyme reactions. So to conclude, in this investigation, the enzyme concentration, if we provide substrate concentration is high, that is, we provide a lot of substrate, and we make sure that the temperature and pH are kept constant, then the rate of reaction is proportional to the enzyme concentration. The more you increase enzyme concentration, the faster the rate of reaction. All right, let's have a look at the investigation here. So in the first question, let's read it together. So it's saying that starch is an important food source that is digested by the enzyme amylase to form the reducing sugar maltose. So we can see here, this is the equation, whereby the enzyme here is amylase. The enzyme amylase will basically help in the hydrolysis of starch. So the word hydrolysis, we use the word hydrolysis because water will be used to break down the starch. So remember, amylase is the catalyst, is the enzyme, and it helps in breaking down starch in the presence of water to give maltose. Now in this investigation, what's the aim? The aim is to investigate the effect of enzyme concentration on the rate of digestion of starch. So we want to find out what happens when we change the concentration of the amylase, that is, we add more amylase or we add less amylase. We want to find out how fast, when you see the word rate, it means how fast the starch is being digested, is being broken down to maltose. So as follows, you have those different steps describing the method for the investigation. Let's read through them and let's find out how this investigation has been carried out. So we can see step 1. It said that 3 test tubes were labeled A, B, C. As you can see here in the diagram, A, B, C, 3 test tubes. And basically in these 3 test tubes, in step 2, 5 centimeter cube of starch solution was put in each of these test tubes. So we add 5 centimeter cube of a starch solution in the test tube A, B, and C. So it's 5 centimeter cube. Next step, we label another 3 test tubes. This time we label them as A1, B1, and C1. And the difference here is we're not adding the same amount of enzyme, but in fact we're adding different amount amylase, the enzyme amylase. Remember, the investigation is about effect of enzyme concentration. So in A1 we had 3% amylase solution. The volume is the same everywhere. Bear in mind, so it's 1 centimeter cube of 3% amylase in A1, and then you have 1 centimeter cube of 2% in B1 and 1% enzymes or amylase in C1. So to conclude, in total we have 6 test tubes, ABC, where you have the starch solution. So this is our substrate, right? So starch is the substrate. Bear in mind, is the substrate. And in A1, B1, and C1, we have the enzyme amylase in different concentration. All right, let's look at the next step. So from step 1 up to step 4, it was all about labeling the test tubes. So in total, we have 6 test tubes. It's about putting the starch solution in ABC and putting the different enzyme concentration in A1, B1, and C1. So the next step is about putting all these tubes, all of the 6 of them, in a water bath at 60 degrees for 3 minutes. Now, why do we do this? We do this in order to equilibrate, all right? So the term is equilibration, to equilibrate all these tubes. The goal is to make sure that the temperature of these tubes, that is the content inside those tubes, the solution that's inside each of these tubes, should all be at the same temperature. So remember, when we do an enzyme investigation, enzymes are indeed affected by different things, right? Different factors. And temperature is certainly one factor that affects enzyme. In this investigation, we are not investigating the effect of temperature. We are investigating the effect of enzyme concentration. Therefore, we have to keep the temperature constant. So that's why water bath is used, to maintain the temperature at 60 degrees. And we keep it 3 minutes to ensure that all the tubes are equilibrated, that the temperature inside each one of them is really at 60 degrees. All right. So that's a very important step in any enzyme reaction. So step 6 now, next question is, all right, so we have the enzyme, we have the substrate. How do we measure the enzyme activity? How do we know how fast or slow the enzyme are working? In this particular case, we're going to use a white tile and iodine solution. So first, the white tile is set up as follows. So as you can see in figure 1.1, we basically divide the tile in 3 sections. So A, B, and C. And you can already guess that section A will deal with the tubes A and A1, and section B will deal with B and B1, and C, section C, C and C1. All right. So in this case, we place iodine solution, drops, if you remember what we've done in the lab. The iodine is placed on the tile like this, so in 10 rows, and put 2 drops, all right, along each rows, all right? So in this case, let's think for a moment, why are we doing this? Number 1, we want to measure the digestion of starch, right? How fast the starch is being broken down to maltose. So we know that iodine, normally it is brown when there is no starch, and it actually turns blue-black in the presence of starch, isn't it? So blue-black in presence of starch. Apologize for the handwriting. It's very, very hard to write with the mouse. Right, so that's basically what we're doing. So we're going to observe, have a look at the tile, and see if the iodine solution changes color, depending on the sample we're putting on it. All right, so this will make a bit more sense, what I've just mentioned, as we look at the next few steps. So we have our tiles, we have our iodine placed on them as shown in figure 1.1. We have those tubes in the water bath. What's the next step? Well, the next step is we have to initiate the activity, the enzyme activity. So basically what we're going to do, all right, a glass rod, remember, it is an enzyme activity experiment. So we basically have to do is to mix the substrate and the enzyme together and allow the enzyme to start breaking down the starch, isn't it? Very simple. So first, before doing this, what we're going to do is we'll do a kind of a check to see when would the iodine change color. So step 8, what it is telling you, it's take a glass rod, place it in test tube A. Remember, what's test tube A? Test tube A has 5 centimeter cube of starch. So take a glass rod, dip it in the test tube A, take a few drops of the starch in test tube A and place it on row 1. Okay, so in the section A on row 1. Obviously, the iodine should turn blue-black because in test tube A you have starch. Then, so we do that. So one time here and you touch the glass rod a second time here. So you should get, should turn blue-black. Step 9 is basically telling you, do the same thing now. So take that glass rod, rinse it, wash it, and basically dip that glass rod in the test tube A1 this time. Now remind yourself, A1 has the enzyme amylase, 3% amylase. So dip the glass rod in it, and then this time place it on row 2. In fact, I have done a little illustration here. So row 2, you're going to take the content from A1, which is basically the enzyme, and touch it here, and touch it here, here, and here. All right? Normally, it should not change color. The iodine should remain brown. Why is it so? Because amylase is a protein. You have absolutely no starch in A1. So this is, so the test, what we've done with row 1 and row 2, it's really to check in which test tube you really have starch. So normally in the A1, there should be no starch because you have only the enzyme. Then the next step is, let's look. The next step, we're looking at step 10. This is where it's very, very important. That's why when you're doing an experiment, you should always read all the steps first. So we're going to start the experiment and we're using a timer. Ask yourself, why am I using a timer? We're using a timer because we are measuring the rate of enzyme activity. So we want to know how fast the amylase is breaking down the starch. We want to find this out. So we're going to start a timer. Now this is where you have to work very, very quickly. As you can see, start the timer. The amylase solution in test tube A1 was poured in test tube A. So this is where you're mixing the enzyme with the substrate. That's where the moment the enzyme gets in contact with the substrate, guess what? The enzyme starts the catalysis, the starch starts getting broken down, isn't it? Into maltose. And this is where it's saying the mixture of starch and amylase in test tube A, so it's all poured in test tube A with a glass rod. So we mix it. We need to make sure that the starch and the enzyme are nicely been mixed together. So we stir the rod, glass rod, and then some of the mixture was immediately, you can see the word immediately has been put in bold. Why is it so? Because it's important. That's where the enzyme reaction, all right, the enzyme starts breaking down the substrate, starts. The reaction starts here. And where do you put that sample? That's your first sample. So your enzyme and your substrate are together, your amylase and your starch are together. We take a drop from that tube and then we're going to place it on row 3. So really, when you're looking at your tile, row 3 is your starting point. Row 3 is where we are starting the measurement for this experiment. So row 3, we place a drop, right, immediately, once the enzyme and the substrate has been mixed. And then what are we going to do subsequently? After 1 minute, all right, we allow the reaction to run. Just remember, once you've mixed the enzyme and the substrate together, okay, you can see here, test tube A, the enzyme, well, it keeps working, okay? If you visualize, you have those enzymes moving around, you have the starch moving around. So those enzymes are breaking down the starch. As time is passing by, more and more of the starch is being broken down. So the logic is over time, as time is passing by, you're going to have less and less and less starch available in that tube. Does that make sense? Because the starch is being broken down by the enzyme. So after 1 minute, the instruction here says, after 1 minute, the glass rod was used to remove some of the mixture from test tube A and touch onto the first drop and then the second drop of iodine solution in row 4. So see what I've done? So on the section A, and the glass rod was rinsed and dry. Remember to make sure that you dry it up. So, well, this is an ATP. You're not redoing the experiment. It's describing it. So row 4, what we know now is on the white tile, row 4 represents, okay, the sample, or represents the reaction after 1 minute. So we dip it, the glass rod here and here, and we see, we look at the color change. Basically then, the next few steps, what it's telling you is repeat that procedure after every 1 minute. So for row 5, it will be the sample after 2 minutes after the start. and then row 6 will be 3 minutes, 4 minutes, 5 minutes, 6 minutes, and the experiment ends at 7 minutes. So we're going to basically observe whether starch is present in that tube, at these different, at these different stages. That is, after 2 minutes, do we still have starch? After 3 minutes, do we still have starch, etc. And guess what? In your ATP, they have given you the results. So the result is in figure 1.2. So figure 1.2, as you can see, section A, the first 2 rows, we don't count them because we know it was mostly just to check whether test tube A and test tube A1, had starch. But however, from row 3, we know we have started the experiment, like the enzyme has been mixed with the starch. And then the subsequent 1 minute interval that we had, just remember, all the subsequent rows are basically showing you the reaction after with a 1 minute interval. As we can see here, I'm going to interpret this first result for you. For test tube A and A1, what we can tell? We can tell that the experiment started, row 3. After 1 minute, we tested the sample, there was still starch. However, after 2 minutes, you can see 1 minute and then 2 minutes. After 2 minutes, there were no more starch. What does this mean? This means that with a 3% amylase, that's a concentration, very concentrated, like with a concentrated solution of enzyme. With a lot of enzymes, the reaction happens very fast, right? All the starch were broken down literally after 2 minutes. Just 2 minutes was enough for 3% amylase to break down all the starch. There were no more starch left in the experiment. Therefore, this is how we have managed to measure the rate of the enzyme reaction, for the test tube A. So for 3% amylase, we know it takes 2 minutes for 3% amylase to break down the starch. Then when you look at the instruction that is shown over there, that is for step 13 and step 14, what it is saying is repeat this entire process for both the test tube B and B1, right? And the test tube C and C1. And as you can see in figure 1.2, which is just here, it's showing you the results for the test tube B and B1 and C and C1. Okay, so using this result, I need you to interpret the results and then answer the questions in that question paper. I hope this video was very useful. Thank you for watching. If you have any question, post your questions in the comments section below. And don't forget to subscribe and like for more videos.